Kia ora, ladies and gents. It's a great privilege to be here, and thank you so much for your introduction. My name's Theo Portlock, and as you said, I'm a research fellow at the Liggins in New Zealand. And today I'm going to talk to you about childhood undernutrition. So undernutrition occurs when the body doesn't receive the nutrients that it needs. Simple as that. And in childhood, this can lead to stunted growth, wasting, and cognitive impairment. And of course, in severe cases, it's fatal, and it contributes to almost half of all childhood deaths worldwide. So undernutrition is a global crisis that's made worse by international conflicts, by pandemics, and of course the cost of living. So really, one of the tricky things about it is that it's this intergenerational self-reinforcing cycle. So what do I mean by that? Well, sort of parents who are malnourished, they struggle to afford nutritious food for their kids. Those kids then go on to become malnourished themselves, and they have deficiencies in their brain development, and that can lead to poorer performance in school, that leads to lower educational attainment and poor economic opportunities in adulthood. And then they become malnourished parents themselves, and so on. So thanks to sort of national and international initiatives, therapeutic foods, or RUTFs, are doing a great job in sort of addressing this issue and refeeding populations that are in desperate need to get their growth sort of back on track. However, the problem with these methods is that they predominantly focus on the weight gain, particularly short-term weight gain, without an emphasis on the neurocognitive development, which is really crucial for breaking this cycle. So, malnutrition impacts a number of bodily systems during development, and one of these is the gut microbiome. We're on MVIF, so I'll talk about the microbiome. So the godfather of a lot of this is a bloke called Professor Jeffrey Gordon and his lab, and he looked at sort of how malnutrition renders the gut microbiome sort of immature, as in is predicted to be much younger than it should actually be, and that immaturity persists even after refeeding. So this sort of got all of us thinking how that persistent immaturity is connected to immaturity of other systems, because development is a system of clocks. Each clock will run at a different pace, and those paces are all intertwined. So background to the study is that in the capital city of Bangladesh, Dhaka, this is a nation that has very high rates of malnutrition. And what we did was we looked at 140 one-year-olds who were malnourished and compared them to a cohort of well-nourished kids. And we recorded all the data across the board, deeply phenotyped from gut to brain to the blood, and lots of socioeconomic measurements as well. So I'll start with the gut. So what we saw was really sort of like what Gordon's lab also saw, that the microbiome was sort of rendered immature by the malnutrition that they were suffering from. So this is characterized by sort of lower diversity or alpha diversity, and a significant difference in their beta diversity, and an enrichment in sort of these oral commensal species, sort of these Streptococcus and Rothia, which are indicators of sort of an immature gut. So next, what we did was we looked into the plasma lipidomics, and we have a guy here, Chris Pook, who did a great deep dive into seeing which lipids were upregulated versus downregulated, and actually it caused a massive deal of difference. So at that 1 year time point, malnutrition was associated with huge differences in plasma lipids, and some of those were more interesting than others. So we have these odd chain fatty acids, which actually that indicates that these are bacterially derived, because we actually only synthesize fatty acids usually an acetate at a time, so every two. So odd chain is sort of an indicator that these could have bacterial origins. The other thing that was missing here are the ceramides. So the blue box here are the healthy, and the orange is the MAM. And we saw that these ceramides and sphingomyelins are important for healthy brain development. They were impacted or differentially abundant between the malnourished and the healthy group. So then, linking it all the way to the top, the brain, we looked at sort of brain function and brain structure. So to begin with, the team over in the Boston Children's Hospital had devised a sort of cap that was able to sort of look at the amplitudes of different EEG frequencies in different regions of the brain. And what we saw was that the amplitudes of the beta and gamma in the temporal and frontal regions of the brain, they were significantly decreased in malnourished kids versus well-nourished kids. And these regions and these frequencies are associated with communication systems or the development of language and communication. And actually then we did a bunch of behavioral tests as well to the kids to see how well their vocalization skills were. And we did that using a system called the Bailey's scales of infant and toddler development, as well as a more local sort of version of this called the Walks test. But they both sort of said the same sort of thing, that vocalization here was being impacted by malnutrition in these 1-year-olds. So we have here a big list of all these things that were different between the conditions, but that's not very useful. The next step was to figure out how all these differences tie into one another. And so to do this, we built an ensemble model that predicted MAM, and it does so very well by merging all the data sets that we had from the microbiome to the plasma lipids to the brain waves. And then by doing interpretation of that AI algorithm, we saw actually some features were more important to the AI. And here is a comparison of the importance of the feature on the y-axis versus on the x-axis, the significance value by mixed modeling. We saw certain species and features that were more important than they assume would be, linearly speaking. So some of those were gut bacteria, and Bacteroides fragilis were among those. So they weren't all necessarily significantly different between the groups, but they were important in the prediction. So then linking all this together, network analysis is a great tool to really understand and build mechanisms from all these underlying changes. So these were the important features for prediction, and they were connected by Spearman correlation. And we saw actually that these changes, the Bacteroides fragilis and its pyruvate fermentation pathways, were connected to higher cognitive function, higher frequency EEGs and expressive communication. And that this pathway was being impacted by the oral commensal species that were enriched during malnutrition. So this is really sort of a full mechanistic understanding of how malnutrition might be impacting the human development, and it offers sort of therapeutic avenues for new interventions that target these pathways and really break the cycle of malnutrition. So I want to thank the MVIF again for hosting me, and I want to thank you all for your time, and I'd love to hear some questions.